Secondly, the rule is you, okay, first of all, yeah, I should have asked this question before. Do you like biology or do you not?
Yeah, I do like violation.
Oh, then good. And half of my work is already done. Anyway, if you have any doubts, you can ask n number of times. Okay. And pay attention because I asked a lot of questions. All right. Shall we start? Okay.
Yes, ma'am.
So, sexual reproduction in flowering plants. Why flowering plants? See, first of all, when we studied about plant diversity, meaning plant kingdom, in that we studied about all the different divisions of the plants. We studied algae, bryophyta, pteridophyta, gymnosperms, and angiosperms, right? Madam, stop. So, talking about angiosperms. See, angiosperms are the only one out of the whole entire divisions of the plant kingdom that are known as the flowering plants. Why so? Is because they produce flowers. And what is the use of flowers for plants? It is their sexual organ. Okay, it is the part through which the sexual reproduction takes place in the flowers. Okay, so this is the reason why sexual reproduction in flowering plants. Okay, flowering plants means the angiosperms or vice versa. Okay, now in this chapter we're going to study all about how the sexual reproduction takes place, what are all the structures that are involved, okay, and what are all the processes that are involved, and lately what are all the steps that go one by one. Okay, now no matter whether it is for angiosperms or whether it is for humans, the sexual reproduction, the whole life cycle can be divided into three phases. Okay, the first phase is the pre-fertilization phase. Okay, the second phase is the fertilization phase, and the third phase is the post-fertilization phase. Okay, now what is fertilization? So fertilization is the process wherein the fusion of the male and the female gamete takes place. Okay, that is fertilization. Now pre-fertilization events are all those events that take place prior to this fusion of gametes. Everything that happens before the gametes fuse are pre-fertilization events. Then comes the fertilization wherein the actual fertilization process is taking place. And lastly, there is the post-fertilization events, meaning all the events or development that will take place after the fertilization has been done. So we'll divide the whole entire sexual reproduction into three phases and study likewise. Okay. Now, talking about the first phase, that is the pre-fertilization phase. Pre means before, right? We know that. Now, everything that happens before the fertilization, pre-fertilization. Now, two things happen in that. The first is gametogenesis. Second is gamete transfer. Two things happen. Okay. Gametogenesis. Gameto means referring to gametes. It can be male gametes, female gametes. Genesis means formation. So, first of all, in order for the gametes to fuse, gametes have to be formed. The first step is gametogenesis. Secondly, when the gametes have been formed, now again, in order for the fusion to take place, the gamete have to be transferred, isn't it? Without them reaching together, how will the fertilization take place or the fusion take place? So two things happen during the pre-fertilization stage. The first is gametogenesis, that is formation of gametes. Then there is gamete transfer. Okay. Now we'll study each of these steps during like in depth. But let's see, during the fertilization stage, what will happen? The gametes that have been transferred, they will fuse. And when the gametes fuse, zygote is formed, isn't it? What is the ploidy of gametes? What is the ploidy of gametes?
Haploid.
Gametes are haploid. What is the ploidy of zygote?
The diploid.
Okay, diploid. Okay, first of all, tell me what is ploidy? Ploidy. Abdul, you can also try. What is ploidy? Ploidy. See, ploidy means the number of chromosomes that are found in any individual. Okay. Now, ploidy can be when one set of chromosome is there, then that is haploidy. That we call it as N, isn't it? Then when two sets of chromosomes are there, we'll call it diploidy, 2N. When three sets of chromosomes are there, we'll call it triploid, right? 3N. And when more than three, then we'll call it polyploid. Okay. So whenever I ask, no matter for which organism, no matter for what, if I ask what is ploidy, what is the ploidy of this particular thing, you'll have to answer in terms of the number of chromosomes. Okay. So just like that, the gametes will always, no matter what happens, gametes will always be haploid. Always remember this one thing. Okay. Gametes means haploid. And when two haploids will fuse, we'll get one diploid zygote. And we can also say that the end product of fertilization or the end product of fusion of gametes is the zygote. Okay. So during fertilization stage, the formation of zygote takes place. Now, after the fertilization has been done, then comes the post-fertilization stage. During this stage, embryogenesis takes place, meaning from the zygote, now the zygote develops into an embryo. And then from the embryo, different organ formations take place, cell division takes place. All of these are the events that take place after fertilization. This much is clear? Any doubts up till now?
Can you
All
explain it again?
Which part? Which part?
Pre-fertilization.
Pre-fertilization or the whole thing?
No, not the whole thing, but like the last part you explained.
See,
I was paying attention.
yeah, see, pre-fertilization, every single event that will take place before the fertilization. Okay, in that, like if I'm putting fertilization as a reference, so everything will be according to fertilization only. Now, pre-fertilization events are all those events that will take place before the fusion of gametes. So first of all, gamete formation will take place. Secondly, when the gametes are formed, they will be transferred. So gamete transfer will take place. Clear? Then comes the fertilization stage. So during that, the formation of the zygote will take place, meaning when two gametes, meaning one male gamete and one female gamete, when they will fuse, we'll have one zygote. And at this point, I asked the question, what is the ploidy of gametes? Tell me, Abdul. The gametes are? Ploidy of gametes?
If it has two
Say.
cells.
Two cells? No. I'm asking about the ploidy. Ploidy. Meaning how many sets of chromosomes it has?
Two.
No. Gametes have one set of chromosome. Gametes are haploid. This is what I was teaching. Please pay attention, okay? Gametes are haploid in nature. Haploid means one set of chromosome, okay? Now, when the gametes fuse, okay, zygote is made. What is the ploidy of zygote? Abdul?
Huh?
What is the ploidy of zygote?
Zygote.
You told me that gametes are haploid, right? Now, a zygote is formed after the fusion of the two gametes. So what is the ploidy of zygote? Tell
Two
me.
chromosomes.
Yeah, you're right, but say the technical term. Give the right term.
The other one haploid and the other one. No.
Diploid. Okay.
Okay.
Two sets of chromosome, meaning one from male gamete, one from female gamete. They become a zygote, and that is diploid. Okay. Are you having difficulty in understanding?
No, ma'am.
Can you please pay attention? Are you writing something?
Yeah, I'm writing this down because if I don't write, I can't like.
Huh?
If I don't write, I can't like get it.
Okay.
I can't get what you teach.
Okay. Okay. All right. So that is the fertilization stage. Then comes the post-fertilization stage. Now, during the post-fertilization stage, all the events that happen after the fertilization has been done, they take place. Meaning, from that zygote will turn into an embryo. Okay. That embryo formation, just like how gametogenesis was there, same way the genesis of embryo will be known as embryogenesis. So embryo formation will take place. Cell division, cell differentiation, different activities will take place after the fertilization has been done, and that is post fertilization stage. Clear?
Yes.
Okay. Now we'll talk about all the structures, all the events that are involved in the pre-fertilization stage. Now, this is a flower right in front of you. Okay. Now, when you look at a flower clearly, now I should have brought a flower. Anyway, I don't have it right now,
I
but
didn't write.
I'll give you time to write. Just let me finish giving the introduction, then you people write. Okay. Now, see, when you look at a flower, what all things do you see in it? Mostly we see the petals, right? Then we see the lower green part, that is the sepals.
Sepal.
Wait, just 1 second. Okay. Do you see this? I have this dried up flower. Okay.
Yeah.
Are you able to see it?
Yes.
Okay. Now see, even though it's very badly appealing, but do you see the petals?
Yeah.
This is what we see from the outside, right? Look at the screen. And these little things, have you seen this? The green ones that are right below? These things,
Sepal.
right? See, right? These things, these are the sepals. Okay. Now you might have seen, now this is a very dried and crunchy flower, but you might have seen that whenever a flower blooms, it has a very bulgy structure here from which the petals come out.
Thalamus.
That is actually the thalamus, right? You might remember from morphology chapter,
Yeah.
isn't it? So now when we go inside this particular part, okay, right inside, I don't know if this flower has it, but when we go deep inside, right in the middle of this, we'll find the male and the female parts of the flower. Okay. Now, talking about, see, if we, like how when we studied about our skeletal system, we divided our skeletal system into axial and appendicular. You remember that?
Yes.
Abdul, can you pay attention? Please look at the screen. So yeah, I was saying that when we studied about our skeletal system in class 11, so at that point of time, we said that there are two parts, right? One is axial, one is appendicular. Same way, when we talk about a flower, for our ease of understanding, we'll divide it into certain sections so that it is easier for us to study. So we will divide one flower into 4 whorls. Whorls are, you can call whorls as the parts of a flower. So 4 general parts. Okay. There is androecium, there is gynoecium, there is calyx and corolla. Now, androecium and gynoecium, these two together are known as reproductive whorls.
Essential.
Yes, they are the most essential. And then these are the accessory whorls, right? Now, what do I mean by accessory is that they are not directly participating in reproduction, but they are helping the reproduction in, like how there were accessory pigments, right?
Yeah.
They help the process of photosynthesis. Same way here, the accessory whorls will help the process of sexual reproduction. Now, coming on to here. So the reproductive units of a flower is androecium, which is the male unit, gynoecium, which is the female unit. Then talking about the accessory unit, we have calyx and corolla. Okay. Now, each of these units will have one single unit. For example, if I'm saying androecium, which is the male reproductive part, just like how in chemistry we have polymers. You have studied polymers?
Yeah.
One
Monomer.
polymer is made up of several monomers. Same way, imagine these as polymers only. They are a larger unit, like one male reproductive unit is androecium. But if we want to find out one unit of androecium or one single unit of, you can say one monomer of androecium, so the monomer of androecium will be called as stamen. Repeat after me, stamen. Say.
Stamen.
Same way, the gynoecium will also have one single unit. So one single unit of gynoecium is carpel or pistil. It has two names. Okay, carpel or pistil. Okay. Then one single unit of calyx, the green portion that was right there, it will be sepal. Okay. And then one single portion of corolla will be petal. Okay. So the colorful one, the beautiful one is the petal. The green colored one, like tiny petals, are the sepal. So single units of them, we'll be talking about that only because, you know, when many stamens come together, we'll call it as androecium. So we can't study 10 things at once, right? It will be complicated. So we'll take one to study it in depth. Clear? So
Yes.
we have single unit of androecium, that is stamen, single unit of gynoecium, that is carpel or pistil, single unit of calyx, that is sepal, and single unit of corolla, that is petal. Clear? This much is clear?
Yeah.
All right. Now you people can note down nicely. Draw this diagram. Okay, it's important so that you can identify each of these things. All right. Is my the handwriting is properly visible?
Yeah.
Yes, ma'am.
Okay. Now you can note it down. You should also note this. Ploidy means sets of chromosomes. Just one second. Also, do you people remember that out of all the divisions that we have studied, algae, bryophytes, gymnoterido, everything, is there any one amongst them who makes flowers?
Angiosperms.
No, apart from angiosperm?
No.
No, none of them. None of them have flowers. This is only the unique characteristic feature of angiosperm, purely. Okay? Now you can note down.
Done, ma'am.
Both of you are done?
No, I need a minute.
Okay.
You can scroll down. I finished till the during the correlation. Yeah. Do we have to draw the diagram?
Yes, draw the diagram.
Done, ma'am. Done, ma'am.
Are you done, Abdul?
Yes, ma'am.
Okay. All right. So now moving forward, we'll talk about the stamen, that is the male reproductive unit. Now, stamen has two parts. Okay, one is anther and filament. So anther and filament together make up one stamen. Now, this structure that you see right here, this lobular structure, this structure, right? This part. This is the anther, and this long part that you see right here is the filament. So two parts make up one stamen. Now, talking about the filament, so filament is a long and slender. Slender means thin. So a long and thin stalk-like structure, a stick-like structure that holds up the anther is the filament. And the proximal end of the filament, see, in terms of biological distances, proximal means front. Okay, distal means back. Okay, so proximal end, meaning which is the front end of the filament, it is attached to the thalamus, that basal bulgy portion, or it can be attached to the petal. This part. Okay, this part, the lowermost part will be the proximal part, because this is the part that will be attached either to the thalamus or to the petal. And the part that is attached to the anther, this part is the distal part or distal end. Okay, so two ends are there, proximal end and distal end. Distal end is the back end or the last end. So the last end is attached to the anther. The proximal end, the proximal meaning the first portion, it is attached to either the thalamus or the petal of the flower. It can be attached to either of them. Okay. Now, see, it depends from flower to flower how many stamens could be there and what will be the length of the stamen. For example, if it is a huge flower, then obviously the stamen would be huge, like it will be very long. But if it is a smaller flower, then why would the stamen be longer than the flower, right? The stamen will also be short. So it depends and varies from flower to flower. We cannot measure it as one general term, okay, because it varies from one flower to another flower. Okay, now we have discussed about the filament. Now let us discuss about the anther. Now see, anther is bilobe. Bi means two, lobe means lobe-like structure. So two lobe, it like one anther is made up of two lobes. So we'll call it as bilobe. Okay, and it is dithecous. Di is again two. Bi is two, di is two. Thecous means theca, a layer. So two layers are there and two lobes are there. So one anther is bilobed and dithecous structure. Okay, bilobed and dithecous. Now, a longitudinal groove separates the two theca. Okay,
What is theca?
theca is layer. The two layers that is forming the anther, that is the theca. Okay, now when we cut, see, do you see this line right in the middle?
Yeah,
This portion?
yeah.
Okay, this is that longitudinal groove that is separating the two theca together or two lobes into two halves. Okay, now when we cut a transverse section, see what is a transverse section? Initially, if this two structure has been given like this, if you join it like this, and then a cut was made in the middle and then it became two, like how we crack an egg, right? This part, see this,
Yeah.
we are talking about the anther only. This structure is the anther, the yellow part. Okay, so here this anther, if we take it and we cut it like this, okay, then that is a transverse cut. This cut is transverse, this cut is longitudinal cut. Meaning a vertical cut means longitudinal, a horizontal cut means transverse cut. Okay. Now, for example, if we cut like how we crack an egg, okay, when you crack an egg, if you look at the two halves, so one is like each of those, if you like, for example, if this is an egg, you crack it half, you have these two portions. Now, when you look at like this, when you look it towards yourself, then it would appear circular in outline, isn't it? Same
Yeah.
happens for here also. Okay. Okay. So when we cut a transverse section of an anther, we can see that the anther is tetragonal structure, not tetragonal. When we like in that circular portion, we'll find that the anther has 4 different columns present. See the columns that are shown right in the figure. Do you see? 1,
Yeah.
2, and then 3, 4 at the back. Okay, so 4 different columns will be there. So because of which we will call the anther as tetragonal. So now we have
It was bilobed, right? So there should be two.
exactly, but each of those 2 lobes have 2 structures present. Bilobed was there, 2 lobes came out.
Yes.
This has 2, this has 2. So when we join them, it is tetragonal. Okay. So now in totality, if we talk about anther, it is bilobed, it is dithecous, and it is tetragonal. 3 properties, very important. Okay. Bilobed, dithecous, tetragonal. It always comes in question. Okay. Say, were you asking something? All right. So, yeah. So when we cut a transverse section, now you understand what is the difference between transverse and longitudinal section?
Yeah.
So we found out that there are 4 sides in the anther inside. Okay, we call it as tetragonal structure. Now those 4 ball-like structures, do you see those purple ones?
Yeah.
Right? It consists of 4 microsporangia. Those ball-like structures are microsporangia. So when we cut inside the anther, we found out that there are 4 microsporangia, and each of those, like 2 in each of the lobes, right? 2 in one lobe, 2 in another lobe. So now we find out, and see, I've drawn this for a better understanding. Like 2 lobes are there, right? One is in the upper part, one is in the lower part again. So this is how the microsporangia are arranged. And because of this very reason, the anther is known as tetrasporangiate. Also, tetrasporangiate. Tetragonal, it was there. Now tetrasporangiate. Why? Because 4 microsporangia are there.
Tetragonal is the shape, right?
Tetragonal is the anther structure when we cut a transverse section. We find out that there are 4 sides to it. That is why we are calling it tetragonal. Now, each of those 4 sides have microsporangia present. So, 4 microsporangia, so we'll call it tetrasporangiate. So, these 4 qualities define an anther. That's all there is to know. Bilobe, dithecous, tetrasporangiate, and tetragonal. If you miss out tetragonal also, bilobe, dithecous, tetrasporangiate, these 3 are more than enough to describe the angiospermic anther. Clear?
Yes.
Any doubts up till now?
No.
What about you, Abdul? Any doubts?
No.
Okay. What happened? You switched off your video?
Okay, I didn't see it.
You know, no doubts?
No.
Okay. Now see, these 4 microsporangia that we were talking about, that we just saw, these will only mature and develop and become a pollen sac. Okay. Like after they develop into a pollen sac, they will make the pollen grains. Right? Pollen grains are the male gametes of plants. Right? So the microsporangia, from being microsporangia, it will develop into pollen sac, and these pollen sac will start producing the pollen grains. Okay, and the pollen grains are the male gametes in plants. This much is clear?
Yes.
Do you people have any doubts?
No.
Now let us move forward and we'll talk about the structure of microsporangium. Now imagine how inside we are. We started with a flower, we went on to the male reproductive part, then we went on to the single part of that, Then we went on to stamen, right? And then we went on to anther filament. Now we are inside and inside. Now we are inside the microsporangia. We are in that level. Now, after we cut a transverse section of the microsporangia, okay, microsporangia, which was there, the purple circle. Now we are cutting a transverse section of that. Again, we'll find out an oval outline. Now let us see what is present inside that. So inside that structure, this famous diagram that you might have seen in, oh, you may have not seen because you don't have the book, but inside that we'll find 4 layers of wall. Okay, like how cell wall was there, plasma membrane was there. Same way here also layers are there and they have special unique names. Okay, new names. So the microsporangia is obviously, it appears circular in outline, because again, whenever I talk about transverse section or any outline appearing circular, just try to remember the egg example. You crack an egg, 2 portions are there, and when you look at it like this, you'll find it a circle. Okay, always try to remember it like that. So it won't be difficult. Now, that microsporangia has 4 walls present. Okay, now naming it from the outside to the inside. Obviously, if walls are present, one wall will be outermost, then another will be right inside, then another will be right inside, right? So now the outermost wall is the epidermis. Okay, outermost wall is the epidermis. Under the epidermis is the endothelium. Under the endothelium is middle layer, and then innermost is the tapetum. Okay, tapetum, whatever you want to call it. Okay, so epidermis, endothelium, middle layer, and tapetum. 4 wall layers are present where? Inside
the microsporangia.
Microsporangia. How many microsporangia are found in the anther, Abdul? How many microsporangia are there? Say. Yes, Abdul, say. How many?
4.
Yeah, 4. Okay. Now talking about the functions of each of these layers. Now, since 4 layers are there, then they must like have some purpose, right? Something to serve. So talking about the function of these layers, so the first 3 layers, epidermis, endothelium, and the middle layers, they are responsible for protection. The first thing, protection. Second, they help in dehiscence of anther to release pollen. Okay, I'll tell you. Dehiscence of anther to release pollen. Now see, what happens is that, for example, if this is the filament, this is the this thing, anther. Okay, now inside the anther, pollen grains are made. Now when the anther becomes very mature, pollen grains are done being made. A lot of pollen grains are made. Then what happens is that this anther bursts, because of which the pollen grains are spread out everywhere. This property of the bursting of anther so as to release pollen grains, it is known as dehiscence of anther. Dehiscence means bursting. Okay. So apart from providing protection, the first 3 layers helps in the dehiscence of anther. The bursting of anther. Why is the anther dehisced or burst? Why does it burst?
Because the pollen grains are made inside.
They have to be released. Right?
Yeah.
See, like how humans do have a proper system, a reproductive system, right? There is a way through which the male gametes will be transported, but in plants there is no such organ like humans, right? They're very different. Of course, they are plants. So there are so many intelligent mechanisms in plants, such as if we want to make one pollen go to another flower, we burst it. With the energy of that bursting, it will be spread everywhere so that it will reach the plants. Okay. So the first 3 layers have these 2 functions, protection and bursting. I mean the help in the bursting or help in the dehiscence. The last layer, that is the tapetum. It is primarily responsible for providing nourishment. Okay, it is responsible for providing the nutrition, the nourishment to the pollen grains that are developed. Because pollen grains, since they are the male gametes, they require the nourishment and everything that will be provided to them by the tapetum. Okay. Now you people can note down. This much you people have understood?
Yes.
Okay. Draw this also and note down. Are you asking something?
So, microsporangia, it develops into
Pollen sac.
pollen sac.
Inside the pollen sac, pollen grains are made.
Okay.
There are more cells. We will talk about. I'm not revealing all the cells right now. We'll reveal it slowly. But for now, microsporangia will develop into pollen sac, and then inside the pollen sac, multiple thousands of pollen grains will be made. Okay.
Do we have to draw this also? I'm finished.
Done.
Yes, ma'am.
Done. Done.
Ma'am, can you go a bit up? Yeah. Enough.
Done, ma'am.
Okay, listen, let's just keep it till here only for today. Okay, you have to write from here, right? Structure of microsporangium. Okay,
I
I'll
wrote
give you
that.
time. Okay, uh, we'll continue the rest in the next class, okay? Because I have another class to take. Okay,
Okay.
so do you people have any doubts?